Hello all and welcome back to this second lecture on the topic Introduction to Ecology. In this part we are going to study the methods of vegetation sampling. So first of all, what do you mean by vegetation? Vegetation, it is the presence of different plant species at a specific area, and to study such species composition of specific area is called as the vegetation studies. The main aim of vegetation studies is to know about the species, their number, abundance, density, changing composition, mathematical and graphical representation. So, when we study a specific area which has a specific type of vegetation, that study is called as the vegetation sampling. So, after studying the vegetation, we can uh, tell about the number of species which are present in that area, what is the abundance of that species, what is the density, and what are the changing components of the specific area. So this vegetation studies, it gives us a measure of changing biodiversity composition of any area over the period of time. So whatever changes are taking place at a specific area in the pattern of vegetation can be studied with the help of vegetation sampling. So this change, obviously, it may be good or it may be bad, and this can be better understood with the help of vegetation studies. So for studying the vegetation, different sampling methods they can be used, and for these studies, depending upon the type of the data we need and the size of the sampling site and the number of available workers. So there are various methods for studying the vegetation sampling, out of which the first method is the quadrat method. The quadrat is a square sample area of different sizes which is marked off in the plant community for the purpose of vegetation studies. So it may be a specific area in the vegetation which is marked, and especially it is square in shape. Then this method, it is effectively used to determine the exact differences or similarities in the structure and component between the two or more plant communities of related or unrelated vegetation. So whether the plants present in that specific area, they may be related to each other or they may be unrelated. There may be one or two specific plant communities. So the exact differences and similarities they can be studied by using this method. Then the size of the quadrat, it depends on the items to be measured. So whatever number of plants we are going to measure, depending upon that, the size of the quadrat, it may vary. Then if the plant number per unit area is to be measured, then the plot size should be large enough to include significant number of individuals. Okay, so if we want to measure how many plants are present per unit area, so for that purpose the size of the plot, it should be considerably large so that maximum plants they can be included. But care has to be taken that it should be not so small that the plants they cannot be separated, counted, or otherwise or they should be counted without duplication or omission of any individuals. Then the large quadrats with many plants, it may require two people to obtain accurate census, while one person may be sufficient for the smaller plots. So depending upon the size of the quadrat, the number of persons, it will also vary. Then the cover, density and frequency, these are the important aspects of the plant community which can be measured by the quadrat sampling. So in this quadrat sampling, there are basically four types, out of which the first type is the least quadrat method. So in this least quadrat method, the enlisting the names of different plants is undertaken, so that we just take the record of, or we just list down the names of the plants which are present within that quadrat. The second method is the list count quadrat method. In this method, it records the number of individuals of each species represented in each quadrat. So depending upon the number of species which are present, and in each quadrant the list and the number is counted. Then the third method is the chart quadrant method. This method records the position and areas covered by branches, mats or tufts of grasses, mosses, etc. on the graph paper. And these graphs, they are very helpful to compare any changes in the structure of community in the future. So a comparative study of a specific area over the period of time can be measured with the help of chart quadrant method. And the last and the fourth type of quadrant method is the clip quadrant method. In this method, it is basically used to study the biomass or weight of each species. All the individuals are uprooted and its fresh weight or dry weight it is recorded. So these are the four types of quadrants. So in the pictures you can see a square marked in a sampling area, so that the marked square is called as the quadrant. It may be it may be made up of what we can say ropes or either sticks or any such material. Sorry. Once the data is obtained from the quadrant studies, it can be used to calculate frequency, density, and abundance of a particular plant species. So, to calculate the frequency, abundance, and density, we need to collect the data by the quadrant method. Then, once the data is collected, using certain formula, we can calculate the abundance, density, as well as the frequency. So for frequency, the formula is number of sampling units in which species occurred divided by total number of sampling units studied multiplied by 100. Or you can also say number of quadrants in which species occurred divided by total number of quadrants studied multiplied by 100. The value it will give you the frequency. Then for density, the formula is total number of individual of species in a quadrant divided by total number of quadrants studied. So it will give us the density. And for abundance, the total number of individuals of the species in all quadrants studied divided by number of quadrants in which the species occurred. This will give us the abundance. So using this formula, we can calculate or measure the frequency, density, as well as the abundance of a specific species present in the quadrant. Then the second method to study the vegetation is the transect method. In this method, the vegetation to be studied is along an environmental gradient or ecotone. For example, from tropical to temperate regions, from the areas of high to low rainfall areas, or the precipitation gradient, adjacent areas with different types of soils, etc. So all these they can be studied with the help of transect method. In the transect method, a line is laid down across a stand or several stands at right angle. This method of linear sampling of the vegetation is called as transect. This transect method, it is also of two types. The first one is the line transect method, whereas the second one is the belt transect method. So we'll start with the line transect method. In this method, the vegetation is sampled over a line without considering the width. So specifically in this, what is considered? So it is just a single line which is considered for the sampling. Then this line, it is laid over a vegetation with a metric steel tape or steel chain or a long rope, and it is fixed with the help of the hooks. So as you can see in the picture, this is the steel plate or the metric steel tape which is laid across a vegetation, and it runs from one point to the other point. So while laying down this line, The line touches some plants on its way from one point to the other. So you can see there are certain plants which touches this line from its one end to the other end. Then from the observations, in a number of such parallel transects, one can comment on the habitat and other environmental conditions of different portions of the transect. So this is just the one transect that we have made. Similarly, many transects parallelly can be laid down, and the number, or one can comment on the environmental conditions of that specific region. These comments, they can be made because every species has its own ecological amplitude and tentatively expresses the status of available water, edaphic conditions, atmospheric humidity, availability of light, grazing, and other biological pressures. So you can see, for example, here are two types of grasses which are shown in the picture, which touches this tape. So first one is the cover grass and second one is the fork grass. So you can see you have to measure the distance on the scale of the same plant. So here, for example, here it is 1 centimeter. Here the same species, it covers an area of 1.7 centimeters. Again 0.6 and 0.8. So how we are going to calculate the percentage of cover grass? So it is 1.0 plus 1.7 plus 0.6 plus 0.8 divided by 10 units total. So how many are the total units? These are 10. So divided by 10 total units, it gives us 4.1 divided by 10 into 100, that equals to 41%. So that 41% is occupied by this cover grass. So you can try it out for the fork grass as well. So what is the formula? It is percentage cover of species A. For example, cover grass is species A. It is the total distance of species A divided by total distance of line. So this is the total distance of the plant or the species, and divided by total distance of the line multiplied by 100. It gives us the percentage cover of a specific species. Then the second method is the belt transect method. The belt is a long strip of vegetation of uniform width. The width of the belt is determined by the type of vegetation under study. So in close herbaceous vegetation, it is usually 10 centimeter, but it can vary from 1 to 10 meters in the woodlands. Then the length is determined according to the purpose of the study. A belt is usually studied by dividing it into equal sized segments. The length of each segment is generally equal to the width of the transect. So again here you can see in the picture, a line is laid down, and besides this line a quadrant is placed touching the line. So such type of vegetation sampling is called as the belt transect method. These segments, they refer to the quadrant. So you can see all these segments, vertical as well as horizontal, so they represent a quadrant. Then this method, they are used to determine and understand the gradual change in abundance, dominance, frequency, and distribution of different species in the transitional region between two different types of vegetation. So when an area possesses two different types of vegetation, the transitional zone or the transitional region, in this transitional region, the abundance, dominance, frequency, and distribution of the different plant species can be determined with the help of the belt transect method. Then the third vegetation sampling method is the plotless method. So as the plants, they are fixed at one place, plotless sampling methods can be used to estimate density, species composition, growth, and environmental factors. This method, it is mainly based on random selection of points within a particular survey area and can be summarized into four main techniques. So to study vegetation of a random area, this particular method it can be employed. So out of this, the first one is the closest individual method. In this method, the distance is measured from one random point to the nearest individual. So any random point is selected, and from that point the distance of its nearest individual is measured. So this is called as the closest individual method. Then the second method is the nearest neighbor method. In this method, the distance is measured from an individual to its nearest neighbor. So instead of a random point here, an individual specimen or plant is selected, and the distance from that plant to its nearest neighbor it is measured. Then the third method is the random pairs method. In this method, distance is measured from one individual to another on the opposite side of the sample point. So, in this, again the distance of one individual from another, but which is located to the opposite side, is measured. And the fourth type, in this, the point centered quarter method, the distance is measured from the sampling point to the nearest individual in each quadrant. This is commonly used in conducting forest inventories in moderate to dense stands of woody plants that are shrub size or larger, and it is also applied in bunch style garden grasslands. So again, in this method, what is done? Distance between the sampling point of the nearest individual in each quadrant. So if we have many quadrants, then the distance between from the sampling point to the nearest individual is calculated by this method. Then, from the mean value of each of these measurements, the mean area and thus the density of the species can be calculated. Formula for density is D is equal to A upon D dash square, where D is the density of individual or of a specified area. A is the specified area, which may be 1 meter square or 1 hectare, etc. And D is the average of four distances measured from center point to nearest plant in each quarter. Then the units of A and D must be the same. For example, if A is selected in meter square, then D should also be expressed in meters for calculation. Then the same plants are identified, sample plants are identified, and some measure of dominance is taken for each, providing estimates of total and relative dominance and frequency by species for the sample area. Plotless sampling is a sampling method without the use of quadrats or transits. So it is a random method in which the quadrats or transits they are not used. Then this method it is often used for surveys of forest vegetation, especially where a rapid inventory is required. So while surveying, taking the survey of a specific forest area, this method it can be employed. So here we come to the end of this second part. We'll continue in the third part. Thank you.